just to give you a sense of what we've talked about, we've talked about, alignment, calling variance, and then some of the sort of functional genomics. And now I really want to kind of get into the machinery of how people do mining all this information. How do they, put it together and analyze it? And that's, of course, all these data mining techniques. And first, we're going to talk about what's called supervised data mining and then unsupervised. So I'm just going to start off with an overview of supervised mining. And this starts with this picture. I think it's a nice picture from this scikit site that kind of gives you just an, it's like a decision tree of like, what type of data mining should I use? And I think this is nice to look at, so it's sort of, well, do you have enough samples to do something? No, you don't, you need more data. And then, I think one big question to think about is, does your data have labels or not? I think that's a very important discussion to think about, because if your data has labels, you move into what's called this supervised area, and we'll talk about it. And then the supervised data, you can have a kind of classification types of things, and there's different classifiers that we'll talk about and so forth. Then there's other methods where you don't have labels, or clustering methods. We sort of started to allude to them, or we'll talk about them more and so forth. There's also a question of if you have kind of categorical data versus not. There's a discussion if you maybe should be looking at kind of more of a regression problem when you have kind of continuous value data versus a categorical type of data. So these are some of the big divisions people make. So now what I would like to sort of do is try to kind of abstract this all into, we're looking at a table. And if you think about it for a second, we talked to you earlier about how all the database stuff is basically comes into building tables, right? So if you build a proper database, and particularly if you put it into these normal forms, you essentially just end up with a set of tables, right? And a lot of these experiments, in the end, what happens in bioinformatics is you reduce everything to a nice little neat table, okay? And usually, often these tables are 2D tables, they could be 3D tables, but so forth. And let me talk about, just for simplicity, a table. I'm going to talk about tables where I'm thinking about somehow one of the axes in the tables is genomic coordinate. I mean, it could be bases on the genome, genes, and so forth. And then there's another coordinate that's sort of assays or things that are happening. So you could imagine these are different factors binding, different levels of expression in different tissues, whatever. You'd get some big data matrix. And so this is a kind of typical type of thing that you get in bioinformatics. So this could be, if you were doing an experiment that wasn't looking at the human genome, say this was looking at genes in the fly. So you would have genes in the fly, and then maybe RNA in different tissues, you'd have the fly in different conditions. You could phrase it that way, but often, almost invariably, the results of the experiment turn into these data matrices. And so what do people do with these data matrices? Well, like I said, first of all, they think about, do they have a notion of a one of the rows? Well, rows on the previous one are now columns on this. This one, unfortunately, is transposed. Is a labeled response. So you could have a situation where you have different readouts, different genomic positions, and that you're trying to say, is this in class A or class B? Now, this, for instance, could be, am I an enhancer? Am I not an enhancer? That could be one thing. This could be, is this gene associated with, if we're looking at expression, is the gene associated with, I don't know, cancer or something like that? Yes, no, that could be the response here. We have levels of gene expression in different tissues. So again, very general type of thing. And what people often do is they think about this table as representing data in some very high dimensional space. And so the idea is to think about, so for each of the rows, you can think about it as kind of the columns as being kind of coordinates in a very high dimensional space. And then you can think of looking in that high dimensional space at a kind of 2D projection of your data. And so this is a very typical type of thing. Here's a picture of maybe a data cloud in high dimensions where we're looking at this projection from a data table, okay? And if we have labels, we might imagine taking that special column that's the labels and say coloring things. And so we have the greens and the reds and so forth. And so we can imagine labeling things in that particular way. Now, you know, an unsupervised method might just kind of look at how close things are in space together. You know, they might sort of try to cluster the points. Now, obviously, we're looking at a particular projection here in the real space that these points live in, which might be very high dimensional. These things which superficially seem close together here might not really be close together. But the notion for a lot of unsupervised things is to kind of find regions, highly populated regions in this, this data cloud. And often if you have labeled data, you can turn your unsupervised thing into something that's kind of predicting things by guilt by association. If you have a few unlabeled points in an area that's very dense or very filled with other points that are labeled consistently, you might use this very simple like guilt by association approach, which is a very common approach for predicting labels. Now, another idea people have is to kind of divide up this high dimensional space. And, you know, the different methods people have, you can think of like a dividing plane in the space and, you know, say, oh, on one side of the plane, it's mostly this color label, the red labels, and then this is the green labels, and then the unlabeled points we don't know. But of course, we could then, if we built this, if we believe this plane or this hyperplane, whatever, divide space up, we would then label all the other unlabeled points on the green side green and the points on the red side red. And imagine, of course, there's the possibility of misclassification. There could be things that are labeled wrongly and so forth. And of course, you know, that would be one extreme. We could also just make some statistical probabilistic prediction of how likely you're to have a particular class based on your relative position on that dividing plane or so forth, or some other characteristics. So let me talk now about a particular method I'm going to talk about, which is kind of decision trees. And this method uses a very simple approach. What it tries to do is to try to separate the points with the dividing plane that's kind of axis parallel. Now that's a very, as you can almost see from this figure, it's a very simple-minded thing. It's very coarse, but the nice thing about it is because it's so simple, you can kind of repeat this operation in a nice way, and you can kind of divide things up. You can make different cuts. You can cut once this way and divide the data this way, but then you can cut again. You can say, oh, if you're on this side, I'm going to make another cut. And that's basically the notion with decision trees. And now I'm going to say a little bit more about this supervised learning here. So I already talked to you about the distinction between regression versus classification. There's also another notion that people talk about, which we're going to come back to, of kind of how complicated the model is. And people often use the term of regularized versus unregularized, where there's some notion of the complexity of the model being penalized. There's also a notion people like to make between is the parameters of the model, are they explicit parameters that you're refining for the model, or are they not? And they often use the term parametric versus non-parametric. And then there's also a notion of what's called ensemble versus non-ensemble models. There's some very sophisticated machine learning approaches where you actually predict something with a model, and then you maybe use a different model to predict the same thing. And then you have like a hyper model that sits on top of them and combines those two models. And you could think of as averaging the two models, but does something more sophisticated, but kind of puts them together and makes an overall prediction. And people have shown that these ensemble models sometimes work very well. And so that's another distinction. And so now let me drill into one easy to understand method, and that's decision trees. So the decision tree, as we talked about, you can think of it as kind of axis parallel splits. But what the representation that people like to use the most is kind of a tree structure. And here's a picture of a kind of decision tree where, we're trying, this is trying to classify this scene, okay? And it sort of, ask question. You can think of these access parallel splits as kind of asking questions. So this one is sort of asking a question, is the top part blue? And then it's dividing, all the images based on that particular feature. And then is the bottom part blue, asking them and so forth. And as you're asking these questions, you're kind of falling down a kind of classification tree. And so this is, trying to say, are you indoors or are you outdoors and so forth. And the nice thing about this is the questions are, in this particular figure, the questions are kind of intuitive. It's as if you had a kind of, you were querying this, a person was querying. They were saying, oh, well, first I want a clue, is the top blue or not? And the second, is the bottom blue or not? They're almost easy, human, understandable questions. And now one question is, how should you situate your questions and how should you make up rules, okay? And so there's kind of a notion of a rule that is very productive in kind of filtering to your, filtering the classes into two different groups versus a rule that doesn't really help you very much. So if you start off with labeled data, okay, and say you're trying to divide between the greens and the reds, and you ask a question or a rule, I don't know, are you in circles? The rule, it doesn't help you. You just end up with the same, the answer to the rule doesn't really change the proportions of reds to greens. It's not very helpful. Whereas if you have a rule that where with the division of the rule, and I should say these rules are drawn where you can have, they're not necessarily true or false. You would have a rule and you would go into one of three categories, for instance. But you can see in this thing, these categories are fairly pure. So like one path you would go down is all reds, one path you would go down is all greens, and even in this path, you're enriching very much for one color versus the other. So that's a kind of good rule, okay? And so you want to have these type of good rules. And there's many different methods for this. I just want to point out our friend, the information theoretic entropy, which comes up over and over again. People use this type of thing. You want to kind of minimize your entropy to create these homogeneous groups. You can kind of compute the entropy of your group and then see if you're kind of making it more homogeneous, because basically you want to find rules that make your group more homogeneous. And often people use the term information gain to talk about the amount of information theoretic entropy that you tend to gain when you do a split. And then here's a simple kind of algorithm for building a tree. Now, again, people do more complicated things, but just a simple thing is you have many different questions you could ask. You would sort of ask all those questions of the labeled data, and then you would pick the rule that maximizes the increase in the homogeneity, okay, or the decrease in inhomogeneity. And you would then, and after you do that, you would then just go back to your rules and you would do it again. And so you would put your best rules up at the top, and then you would cut down. Now realize, let me see if on the next slide, realize as you're going down one of these trees, a rule that was useless, let's say you had red and green and you dots 50-50 red and green. And let's say the first rule really didn't like one rule really didn't help you very much divide between green and red. And another rule maybe shifted you, say rule 2 would shift you to say 70-30 green red. But when you came back and applied the first rule on now a very pure group of mostly red or mostly green sequences, it might actually be very useful. So the value of the rules will change to some degree on where they sit in the tree. So you can't just sort of say in an absolute way, this is the best rule overall and these are the worst. It kind of depends on the subset they're looking at. And here's a particular example of a decision tree. I thought I would walk you through this. This is a decision tree that was built to classify sequences for their suitability for purification, just some particular type of thing. And what it did is it looked at sequences and it tried to say, it knew that, say at the beginning, maybe it was like 140 to 210, maybe two to three sequences were not suitable, okay? And then it tried to kind of apply rules to find very pure groups of sequences that were very suitable. Now, first rule, it said, is there a hydrophobic stretch? And then, yes, no. And then you can see, just looking at these oval, it would say enrich for, say, the no group here, okay? And then this next rule, I think, was looking at, say, how much of a particular type of amino acid you had. And you would go down, you could see you enrich a little bit more until you would enrich to a very pure subset here and so forth. But notice you can take a different path here and you might reuse the same rule in different contexts. So this rule, for instance, was looking at the amount of charged amino acids, that's D and E, in the sequence. Okay, so after you split down here, this was actually a very useful rule, but actually it turned out it's also a useful rule in a very different context after you followed a different path. And actually, the same rule that you used initially to split things up, you might want to come back on that rule and resplit, use it again. And so that's an example of a decision tree. And this just kind of shows, it just shows in diagrams out when I kind of said how this works. You start off with the number of sequences, and they're grouped into two categories, and you apply this particular rule. Now, one of the issues that we're going to talk about a bit is this notion of overfitting, and we're going to talk about that a little bit rigorously in a second, but I'm just going to kind of pre-introduce this idea. So one of the issues right off the bat is that you can imagine growing the tree, so it gets bigger and bigger and bigger, and until you get completely pure leaves at the bottom, but you've added so many decisions onto the tree. And so the question becomes, is that all useful, or are you kind of, people use the term overtraining the data. And so there's a big discussion of how you cross-validate these trees, and we're going to talk about that in a second. There's also the notion of doing an ensemble of these decision trees. So you could imagine kind of randomizing this type of thing, these kind of divisions, and making one group of decision trees and then making another group of decision trees and then kind of ensembling them together. And people, this actually turns into a very common algorithm that people use, which is called random forest, which is one of the more popular algorithms that people use.